This research is really significant because it demonstrates that there is an alternative mode of action for the treatment of synthetic opioid toxicity.
Hello, I'm Emily Schneider from the American Chemical Society, and welcome to this media briefing from ACS Spring 2023. There's been a recent push in the US to make naloxone, a fast-acting opioid antidote, available without a prescription. This medication has saved lives, but it's less effective against powerful synthetic opioids such as fentanyl. In an interesting twist, researchers are now looking to the marijuana component cannabidiol, or CBD, as a possible alternative to the popular antidote. Today, a team reports compounds based on CBD that reduce fentanyl binding and boost the effects of naloxone. We're joined today by Jessica Gudorf and Taryn Vasquez-Berger from the Indiana University Bloomington. Thank you for joining us.
Thanks for having us.
Thanks for having us.
Great. So can you tell me what you were trying to find out with this research?
So the problem is that we're losing over 100,000 Americans a year to overdose. And at this point, most of these deaths are due to synthetic opiates. Now, we do have one antidote, as you mentioned, that being naloxone, but that's a competitive antagonist. It competes directly with synthetics like fentanyl. So there's actually reason to think that naloxone doesn't work as well then against highly synthetic opiates compared to other opiates like heroin or morphine. So the question we were then trying to answer was whether we could take a different approach. So allosteric modulators bind to a different site on a receptor, so there's actually no direct competition with fentanyl. So what we wanted to see in the end of all of this was whether a negative allosteric modulator could reverse fentanyl's effects.
So Jessica, why is this an important thing to know?
So Narcan, the golden standard, is actually really effective against overdoses for things like heroin. However, it has been shown to be ineffective against fentanyl class synthetic opioids. And years of research have actually gone into development of a better Narcan. However, they've been exhaustive and really have shown to be ineffective or better than Narcan. So we have decided that, with these synthetic opioids, they're not going anywhere, that we really need to investigate another mode of action or a better route to prevent these synthetic opioid overdose deaths.
How did you get the idea to use CBD as sort of the starting point for finding these antidote alternatives?
So our lab actually has previously published work on CBD as a negative allosteric modulator at a separate receptor, but this receptor is really important for modulating or, bringing about the effects of cannabis. And so because of this, we had considered a very similar strategy for combating cannabis toxicity. So once we realized that CBD was also a negative allosteric modulator, or could potentially be a negative allosteric modulator at opioid receptors, to approach this type of question, it wasn't a big logical leap then.
So then how did you investigate these, Narcan alternatives?
Right. And, surprisingly, CBD happened to be the most promising lead compound for this work. So we then linked up with Jesse and her advisor, Michael, to then synthesize structural variants of CBD to test in a cyclic AMP signaling assay. Now, I will say this is the only time that I'm going to bring in some jargon, but I will pull it back really quickly. So at a molecular level, opioid receptors inhibit the synthesis of a really important intracellular messenger called cyclic AMP. So what we decided to do was we wanted to then target this inverse relationship for our experiments by tagging cyclic AMP molecules with a fluorescent protein. So this protein is going to light up when it is connecting with these molecules, I'll say. So by taking these molecules, this then enabled us to quantify opioid receptor activity in cells. So by then measuring fluorescence intensity of our cells over time with lower intensities of the cells correlating with opioid receptor activation, and then higher intensities correlating with receptor inhibition, we were then able to go through several rounds of testing of these compounds, ultimately testing 50 compounds in this specific assay. And then on top of this, we then had the privilege to work with a third lab who had done some computational modeling of the binding site in question. And so this then may help us to further identify additional compounds down the road with this specific project.
Great. And then what did you learn?
We learned that these compounds definitely work in a cellular model by reversing fentanyl's effect on cyclic AMP signaling, but they also work by synergizing with naloxone to further reduce the effect of fentanyl on cyclic AMP, which was really exciting to see. So then from this, we've actually then been able to identify structural moieties of these compounds that are important for these effects that we're seeing. And in addition to this, we've also been able to identify compounds that are actually much more potent than the lead compound CBD.
So what's the big significance of these findings?
Well, it's actually kind of a it demonstrates a proof of concept that we can use allosteric modulators to help reverse opiate overdoses, whether that's alone or in combination with naloxone, which is the current antidote for opioid overdoses.
Great. And then what is the next step in this research?
So the next step in our research is to test the promising compounds that we found in preclinical mouse model studies. And we're really interested in looking at the ability of these compounds to reverse respiratory depression, because respiratory depression is what causes these fentanyl-induced deaths. Pretty much your skeletal muscles tense up and you can't breathe. So we're really interested in looking at how these compounds can reverse that effect. And actually, currently, Taryn has started on these experiments.
And then when do we think that we could see this in clinical applications?
So it's a little too early to tell because we are just starting preclinical research, but we're really hopeful that just from our research and this proof of concept that researchers are inspired by our work, and future work can get to clinical trials, but it's a little too early still.
And what do you want viewers to take away from this research and this presentation? What's your big conclusion?
As we are hopeful that our compounds could, get to clinical trial or through clinical trial, 90% of drugs actually fail in clinical trial. So we really hope that this work inspires other researchers to take this proof of concept and be able to help us save more lives in terms of treatment of synthetic opioid toxicity.
All right, well, thank you, Jessica and Taryn, for sharing your research with us.
Thank you for having us.
Thank you very much for having us.
Be sure to check out our other media briefings for ACS Spring 2023, which will be posted throughout the meeting at www.acs.org/ACS Spring 2023 briefings.